Thank you very much, and thank you to Emmett, Craig, and the OIS Organizing Committee for the opportunity to speak today. I'm Frances Lazowski with 2020 OptimEyes Technologies, excited to share with you today our novel eye drop formulation that we're initially targeting for dry eye disease. So we're a spin-out company from McMaster University, which is located in Hamilton, Ontario, all the way up in Canada. And we've been working on a mucoadhesive Micelle technology. This is a platform technology that can be used for a variety of ocular applications, but we're initially looking at dry eye disease, particularly with the high-dose cyclosporin. As you may know, a micelle is a polymetric sphere that is able to encapsulate drug in its core. This improves the solubility of compounds, and since it can be formulated as an aqueous formulation, typically decreases irritation in the eye. Unlike other micelle technologies, we've been able to incorporate a mucoadhesive component, which actually allows our compound to bind to the ocular surface. This significantly increases the residence time on the eye, allowing for a more sustained release of the drug. We compared our micelle with this binding moiety to a standard micelle, and after 48 hours, there was twice as much remaining on the corneal surface. By 1 week, this difference increased to 6 times as much compared to a normal micelle, really showing the longevity of our micelle on the surface of the eye. So really, this is about changing the dosing paradigm for this drug in the case of dry eye, going from twice daily to once or twice per week, with a more comfortable drop that increases patient compliance and can improve clinical outcomes. As we know, the dry eye market is growing, and we feel that these patients would greatly benefit from a treatment that has reduced dosing and a more comfortable drop. In order to validate this technology, we've compared our formulation to the market leader Restasis using a scopolamine-induced dry eye rat model. Here we had Restasis dosed twice per day and also once every 3 days. And for our micelle formulation, we looked at twice per day, once every 3 days, and once every 5 days. In order to evaluate this efficacy, we use 2 metrics, the first being tear volume, which starts out around 9 millimeters in the healthy animals and drops to about 6 millimeters once the disease is induced, where it remains for the 14-day trial in the control population. As expected, Restasis dosed twice per day significantly improves the tear volume, returning it close to baseline, as does our micelle formulation dosed twice per day and once every 3 days. Of note here, Restasis dosed once every 3 days is not different from the control group. Similar story can be seen with the staining results, the fluorescein staining results, where the baseline is initially quite low in a healthy animal, and once disease is induced, substantially increases. Again, Restasis dosed twice per day, the micelles dosed twice per day, and the micelles dosed once every 3 days shows significant improvement of returning the levels close to baseline. Of particular note here, the micelles dosed once every 5 days showed a statistically significant improvement relative to the control group, although it wasn't to the same resolution levels as the other groups, but we now have a even higher dose formulation that we think when we're testing right now, and we think we can get a better resolution at these later time points. So really the take-home message from this was that the micelles dosed once every 3 days showed a similar efficacy to twice-a-day Restasis in this animal model. We've also tested the safety and the tolerability of these micelles in both rats and rabbits, and they appeared to be safe and well tolerated. So our approach to trying to commercialize this technology is 2-pronged. The first is to use therapeutics that are available on the market, such as this high-dose cyclosporin, and we're also looking at a once-a-week latanoprost dose. And the other is to look for external collaborations with strategic partners who may have a novel API that could benefit from either this reduced dosing, increased solubility, or this particular profile. Our team is led by myself, a chemical engineer by training, and also Dr. Heather Sheardown, who's here today and whose lab this technology originated from, with a complement of very talented postdocs. We're also fortunate to have some excellent advisors, including Dr. Paul Karpecki, who we've all heard apparently a lot about today, and Michael O'Rourke, who are both here today with us. To date, we've been able to do all of this work on non-dilutive grant funding, and we're now seeking out seed money to put together an IND package for submission to the FDA that would allow us to further validate this technology in the people. So we're happy to speak to anybody at the break who's interested in more information on our technology, and again, would like to thank everybody for the opportunity to present today. Thank you.